The goal of this procedure, commonly known as ChIP-Seq, short for chromatin immunoprecipitation followed by next-generation sequencing, is to determine the genome-wide location of chromatin regulators and chromatin states in the Xenopus embryo. This is accomplished by first fixing Xenopus embryos and thereby covalently linking the DNA-associated proteins to the genome. Next, the nuclei are extracted, and their chromatin is solubilized and sheared by sonication. At this point, the DNA-linked protein of interest is purified by immunoprecipitation and extensive washing. The final step is to create an indexed ChIP-Seq library from the purified DNA fragments. Ultimately, millions of 5' ends of these DNA fragments, so-called reads, are sequenced in parallel, aligned to the genome, and extended to average fragment length. Local enrichment of reads, so-called peaks, are determined to identify all the site-specific DNA occupancies of the protein of interest, and, thereby, expanding the study of its genome-wide characteristics.
Though this protocol can provide detailed insights into the genome-wide chromatin architecture of Xenopus embryos, it can also potentially be applied to other organisms with sequenced genomes.
Begin with the dejellied embryos. Details up to this point are provided in the text protocol. Transfer them in an 8 ml glass vial and wash them with 0.01x mmR. After the wash, fix the embryos with 1% paraformaldehyde in 0.01x